All right, so let me give you a rundown for today, I guess, and next week. So I'm going to wrap up Chapter 3 today. And then Monday, you guys have the college fair. So for those that are still here, or maybe you missed the bus, I don't know. It will be a review day. You can ask questions, or if you want to start on the review packet together, I'm cool with that.
Yeah.
You know, we'll just make it productive as much, as productive as possible. Tuesday, we'll do the liver lab, the enzyme lab, and get it done. Like, read your lab the day before so you know what you're doing. Herman Norcross, I will send it over with Jane. I'll pack it in a cooler on Monday, and then I'm hoping I'll ask her to put it in her fax fridge or a fridge that you have access to, because it's got to stay chilled. So, yeah. And then I was originally thinking exam Wednesday, but I'm worried that you may have to finish up the lab on Wednesday. So should we just do exam Thursday? Because I on Google Classroom, I think I said exam Wednesday and then start a unit, but we don't have class Friday because it's homecoming. So should we just do the exam on Thursday?
Might as well.
Yeah.
Does anyone see any problems with that?
That way we can get a review in case we don't finish the lab. In case we do finish
I guess that's true. Yeah, I think I do have like Kahoot.
Because, like, who's going to be here for the daily college fair other than Brooklyn?
Also,
Morgan, if you
at the
miss
college
bus.
fair, if you're thinking about Gustavus next year, let me know because I get you a $4,000 scholarship. So, yeah. Did you hear that, Herman Norcross?
Yes, we did.
Okay.
Oh, it doesn't even sound happy.
No announcement? Okay.
Pause for dramatic effect.
All right, so yesterday, wrapped up nucleotides, started proteins, almost finished proteins. We talked about the levels of structure in proteins, starting with just the amino acid sequence. And in this diagram, you can see that lysine, like the next amino acid, can be lysine, so you can double up. There's no rules as to which amino acid should come after the amino acid before it. So proteins, because of that, are very diverse. So the primary structure is the amino acid sequence. Then hydrogen bonds are responsible for the secondary structure, where it can be an alpha helix or a beta sheet. So those are the two shapes associated with secondary structure. And then when you get multiple folds and more interactions, hydrophobic interactions, ionic bonds, you get the tertiary structure. And then when the tertiary structure binds or connects up with other subunits, then you get your final kind of functional protein quaternary structure.
Wait, is that a domain or not?
This would be considered like a domain, part of a bigger protein.
Okay. And then what's the thing in the old shaken not stirred? What was that?
That's a motif.
Oh, okay. Yeah, 'cause I thought Mom would talk about that.
So then I talked about motifs and domains because proteins are so diverse, we stumble across proteins and we may not know what they do, but they have similarities due to their structure, so then we can hypothesize what it may do, and then we can test and go in that direction to figure out what it does. So I talked about motifs, where you have this reoccurring thematic event or element in music or design. So like every James Bond movie says, shaken not stirred. Michael Bay has tons of explosions. Christopher Nolan likes to mess with your mind. Alfred Hitchcock has something to do with its triangle theory, so you can look that up. So proteins have motifs as well. I talked about the three: beta, alpha, beta motif. So you have the beta sheet, it turns into an alpha helix, back into a beta. You have the beta barrel. A lot of channels and pores are beta barrels, so we'll talk about aquaporins this semester and the shape of that. And then you have the helix, turn, helix, which would be here. So helix, turn, helix, or helix, turn, helix. Those are the three motifs that you find. And then domains are like bigger functional units. Paragraphs, yeah. So if you thought of amino acids as letters and the motifs as words or phrases, the final picture would be your domain, your paragraph. So here's a motif, beta, alpha, beta loop into the domain, bigger protein. So then I talked about how do proteins fold So they fold due to the properties or characteristics that they carry. So like, is it hydrophobic? Is it polar? But that's just one part of the story. They have to have other proteins help them fold, and so we call them chaperone proteins. So here's this misfolded protein. Chaperone protein binds to it, refolds it correctly, releases it, and now this protein's functional. When a protein becomes unfolded, we call it denatured. It no longer does the job that it's supposed to do. So chaperone proteins help other proteins fold correctly. Yes, Matt.
I remember what I was going to ask, and then I got to this slide.
Yep.
But there was one of the diseases we talked about in Rourke's, and it was like, is it like the mad cow disease or whatever, where the proteins unfold in the brain?
Yes.
They can't be refolded.
They're
Why don't they just inject a bunch of chaperone proteins into there?
You know what? That's a good question. I'm sure it's therapy that you could.
Yeah.
Sure.
But maybe they just keep. Because it says that when they bounce around and then they hit one and it unfolds that one.
Mm-hmm.
So the more they put in, they probably
just be a lot.
roasting all the.
Yep. So then a lot of diseases are because the protein is deficient. So I talked about cystic fibrosis, and so give you a picture. Cystic fibrosis, pretty serious disease. It has a lot to do with mucus buildup in your esophagus and nasal passages. So what happens here is we have a cell membrane, and this is the normal channel. Chloride ions, leave the cell, and these chloride ions break down the mucus. So you can see that there's no mucus here. And then they're like, okay, now if you have a mutation in that protein, maybe the amino acid change, maybe it's misfolded in some way, now the chloride ions just build up inside the cell and they don't leave, and as a result, mucus builds up inside the person. All right, so how proteins become unfolded or denatured has to do with their environment. So in your enzyme lab, you're going to see that. We're going to manipulate temperature, and we're also going to manipulate the pH of the protein, aka enzyme, and see what happens to it. So this is why when you pickle or do salt curing, you're basically keeping microorganisms from growth of food due to change in their environment. Their proteins become unfolded, and proteins do a lot of the work for a cell. So microorganisms can't thrive in that environment. So denaturation, when the environment is reestablished, a small protein may refold into natural shapes. We call this process renaturation. The fact that proteins can spontaneously renature means that tertiary structure is influenced due to the primary structure. So that's it for proteins, okay? And now we're on to our last macromolecule, which is the lipids. So I'm going to talk about the difference between saturated and unsaturated fats, and then the bonds that are associated between those two types of fats, their energy content, and what happens at room temperature or not. And then finally, contrast lipids and starches due to either their solubility in water or how much energy they can store. So there's not a lot of slides left here. So lipids are hydrophobic molecules. Hydrophobic means they don't like water. So that means they're insoluble. Lipids are fats, oils, wax, grease. And one characteristic of lipids is they have this very high proportion of CH bonds, carbon-hydrogen bonds. They form really, really long chains, as you can see in this faded background here. They don't fold up like a protein, but instead spontaneously cluster with other hydrophobic molecules.
What's Homer doing
Triglycerides is what he says. And he's got a remote in his hand and a beer. Oh, I probably shouldn't have this image nowadays. Duff beer.
Looked like he was chugging beer.
Okay. Oh, man. Anyways, fats usually kind of, I'm going to talk about triglycerides, but you can break down fats into two kind of monomers, if you will. Fatty acids and glycerol. So these are the monomers. You put it together, you get your fats, basically. Okay, so fatty acids, long chains of hydrocarbons with a carboxylic acid, COOH at one end, and then you may see a glycerol, a 3-carbon polyalcohol, and many are built with just one glycerol and 3 fatty acid chains forming this triglyceride. So here's what the fatty acid structure looks like. Here's my carboxyl group, my hydrocarbon chain. So this is what it looks like if you were to draw it out on paper. Each one of these points represents a carbon and a hydrogen, two hydrogens bonded to it. So this is just like a simplified way to show this molecule. Here's a 3D model, and then this is kind of what it looks like. Here's what glycerol looks like. You can see the 3 OH groups. And then a triglyceride is 3 fatty acids, 1, 2, 3, and that glycerol group. So then you get this, basically.
So carbons just always have hydrogens connected to them?
Yes, when you see these squigglies, boom, it's hydrogen. Unless it's denoted otherwise. All right, so many CH bonds of fats serve as long-term energy storage. So this is where we're going to talk about the difference between saturated and unsaturated. So saturated means that when I draw this little squiggly line, okay, each one of these is a carbon, and each carbon has 2 hydrogens. It's like it's holding as much hydrogen as it possibly can. So it has all the hydrogen atoms that it can possibly have. Unsaturated comes in when you have a double bond. So maybe I should have drew a straight chain here. So we'll do a straight chain. Let's just look at a 4. Okay, so I have 4 carbons, right? And then for hydrogens, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, now unsaturated, you see a double bond. So maybe I'll put the double bond here. Still have 4 carbons. Now we're going to count how many hydrogens I have. Okay, so I have 4 bonds here, carbons happy, 1, 2, 3. 4 bonds here, carbons happy, 4, 5, 6, 7, 8. All right, so this fatty acid is unsaturated because it's not holding all the hydrogens that it possibly could because of this double bond. So we're short a couple hydrogens. It's unsaturated, or you call it polyunsaturated when there's more than one double bond. So the characteristics between saturated and unsaturated has to do with its shape. So if I'm straight chained, well, that's unsaturated. But now I have a double bond, and it causes a kink. Okay, so you can kind of think of yourself as, part of a fatty acid. You got a polar head, and then your fatty acid tails, which could be your leg. And so if it's straight, it's saturated, but then when you get the kink, it's like this. Okay? So as a result, at room temperature, the ones that have the kink are in liquid form. So all oils are unsaturated. If it's liquid at room temperature,
that's why they say those are like better for you. Right? This is an unsaturated fat.
Mm-hmm. And then if it's hard, well then at room temperature, then we call it saturated. Any questions with saturated versus unsaturated? As much of the hydrogen that it can hold, double bonds, fewer hydrogen. Liquid form, solid at room temperature. No? Okay. So the double bonds and the unsaturated fatty acids change behavior. As a result, you don't get the free rotation effects of the melting point. So it's liquid at room temperature. Fats that are saturated are solid at room temperature. There are other kinds of lipids out there. Terpenes. So you find terpenes in pigments, so chlorophyll, retinol, which is in your eye, even rubber, natural rubber. Steroids are lipids, and the characteristic of steroid is 4 carbon rings, and I have a picture of that 2 slides. It's coming up here, and you'll see that. It's kind of like a mega house with a garage kind of shape there. So steroids are usually responsible for testosterone and estrogen in us. And then we also have chemical messengers in our endocrine system that are lipids. lipids. So some common terpenes you may not know, but the smell of pine, peppercorns, cedar, rosemary, lemon. We've talked about lemon oil. Mince, lavender, orange peel, lemongrass, hops for beer, roses, wine grapes, cannabis.
And these are neither saturated nor unsaturated. They're just a different type of lipid.
They're just a different type of lipid.
So how come they put a lot of those in like essential oils?
Because they actually have medicinal purposes or economic. I would say medicinal. Alright, these are steroids. I know the picture is a little blurry, but you have the 3 hexagon rings, and then the last ring is a pentagon. So that is a characteristic of a steroid. So, I mean, if you know that a steroid is the 3 hexagon rings with a pentagon, you could look at anything and be like, oh, that's a steroid, and not know the name of it. So, alright, one more thing I want to talk about with fatty acids is, notice like this is saturated because it's straight legged, and then this one's got the kink. Well, if you have a lot of phospholipids, which forms in your cell membrane layer basically, if you have a lot of kinks, things can wiggle through and pass through it very easily. So it's kind of trying to find the right balance of it, if you will. So sometimes cholesterol, as you can see here, cholesterol is type of steroid, 1, 2, 3, gets wedged in to kind of help with the structure of it.
Wait, so is that why fat that's saturated aren't as good for you, because you get more cholesterol?
Yes. Yep. So fats are excellent energy storage molecules. If you were to compare it calorie-wise, fats 9 kilocalories, carbs 4 kilocalories. So fats store twice as much energy than carbs. Oh, that's a typo. Most fats produced by animals are saturated. Plant fats, however, are unsaturated, so like vegetable oil, margarine. So a lot of times we have to convert it by just adding hydrogens to it, so you can convert unsaturated to saturated. So peanut butter is artificially hydrogenated because peanuts are plants, okay, and it has oil in it. So yeah, it's artificially hydrogenated.
So it just started as like an oil, and they add that to it.
To kind of thicken it up a little bit. So that's why when you buy natural peanut butter, there's usually a thin, like a liquid oil at the top, like, the organic. And that's because plants produce unsaturated fats. So then if you get like my husband gets the Jif natural, but I still think it's artificially hydrogenated because it's not a very, it's just not really at the top, so whatever. Okay, so phospholipids are a type of lipid that's really important for cells because it forms your cell membrane. And it's a bilayer for cells. The tails interact with each other because they're hydrophobic. Cells are mostly made of water, so where intracellular means this is facing inside the cell. Extracellular means facing outside the cell. So the polar heads love to interact with other polar molecules. That's why they're out and the tails are interacting with each other. So a phospholipid is made up of 3 things: a glycerol, fatty acid, in this case 2 chains, and then a phosphate group attached to one end of the glycerol. So it goes glycerol, phosphate, and then the fatty acid tails.
Don't the little the heads, the circles there, act as like, aren't they like a filter kind of, so stuff can't get in?
Yeah. So like small ions may be able to wedge themselves through, but a lot of times they act like a barrier because they're compact together.
I remember talking about there's some Easter rocks.
So we will talk about that in chapter 4 with cell structure. So that's coming up. So another picture of the phospholipid. Here you can see the head. It's got phosphate group, the glycerol, the 2 fatty acid chains. You can see that this one is unsaturated because it's got the little kink. The this is the polar region, which means it has a charge then, and this is the non-polar region, hydrophobic.
And is it the tails that are? Oh yeah.
Yeah, the tails are hydrophobic. So it has a polar head, that's the phosphate, 2 non-polar tails. So we call this molecule, okay, I'm probably going to say it wrong, but amphipathic, which means that it can interact with both non-polar and polar molecules. This is why like soaps are also amphipathic, because soaps, what they do is they form like a little vessel around grease and oil, and then I just gotta draw a picture. This is not going to do it any justice. So like, let's say I have some dirt on my hand. Okay, so that's like some dirt. Okay, and if I use detergent, will the non-polar parts interact with the dirt, because dirt usually is non-polar, and then the polar heads are on the outside, and then water is polar, so then it interacts with the water and it just kind of rolls right off your hands. And it's all because soap has both positive polar region and a non-polar region. So the formation of cell allowed a lot of chemical reactions to occur, and more efficiently, because you can separate the two out and kind of serve as a selective barrier of what comes in and leaves the cell. So this diagram again showing phospholipid bilayer, zooming in on that hydrophilic head and the hydrophobic tail. Tails interact, head's on the outside. This is what I was talking about. Sometimes it's not a lipid bilayer. So like soaps, you know, they'll form, like fatty acid tails will interact with the dirt that's in the center, and the polar heads will follow water and just go down the drain. All right, so that does it for lipids.
Gotcha.